All right, good. Welcome back. Let's go ahead and get started. Few announcements. So that first homework assignment, it's just 2 questions. It's just hey, let's get everybody registered into mastering. And that is due tonight at midnight. So most of you are, there's still like 10 or 15 of you by my last count that aren't registered. So I'm just trying to encourage you to do that so that you have time to complete all the homework when it is due a week from today. All right, so I intentionally made that first exam a little bit earlier. It's going to cover less material than our other exams, be a little shorter. So right now I'm intending a week from today to have a 50-minute exam and then lecture to you the last 15 or 20 minutes. And I know that's not terribly popular, but we lost a day to Martin Luther King. So, you know, I'm planning on recording that like I normally do, but just kind of have that in your head. There's only 45 multiple choice questions on that exam, and 50 minutes should be more than enough time to do that. So I'm planning on that in person. Now, by my count, about 15% of you are out for COVID. So I'm still planning on being here in person, even if we go higher than that. Hopefully cases will start to, you know, at some point everybody's going to get it, right? So at some point, hopefully those come down. I will look at the numbers before the exam, and if there's going to be a ton of you out, then I might offer an online option for that first exam only, but we'll kind of wait and see. Otherwise, I like to have, you know, people come in and do makeups whenever they can. If you're going to be gone for a long time after everybody takes the exam, then we'll skip it and fill in with the final. Still decisions yet to be made, but so that's kind of what I'm thinking at this point. I will post some practice questions after lecture today when I'm kind of sure where I'm going to cut off that material, but there will be a good chunk of material that I'm going to lecture over on Monday that will be on that Wednesday exam. And I don't really like to do that, but it's just kind of sort of the way it worked out. So more on that Monday where that cutoff will be, but probably get through chapter 3 and not chapter 12. I pushed chapter 12 off homework to the next exam due date. So, okay. Any questions about last time or anything going forward? All right, so where we left off last time, we went through some kind of theory about microscopy, what kind of things that we need to understand to see ourselves, and different kinds of microscopy. Some of that you'll get in lab, especially that theory. And then we started talking about taxonomy. So we'll finish that up today and start talking about structures of cells. Okay, so I was trying to get across to you last time why it's important that we have the best kind of classification that we can for microbes. When something new is discovered, we need to kind of understand that new organism's relationship to organisms that we already know. Of course, Linnaeus was kind of the first one that started organizing the scientific community around this back in the mid to late 1700s. And we've been revising it ever since. And we will continue to do that as we get more information. Historically, we've been classifying organisms based on their shared characteristics. Things that we can easily see. And we're going to talk about some of those characteristics in a minute. But understand now we are placing a little bit greater emphasis on phylogenetic relationships. And that means evolutionary. So we're looking at the closest common ancestor to understand how organisms are related. And that really came about due to the work of Carl Woese, George Fox, who we usually give credit as well because he was in Carl Woese's lab. In the 1970s, they were doing work where they were sequencing ribosomal RNA. So ribosomal RNA of many, many different organisms in an effort to try to understand relationships between organisms. And why did they use ribosomal RNA? Why didn't they use DNA? Well, ribosomal RNA, where do we find that? We find that in ribosomes. Okay, no surprise. We're going to talk more about ribosomes probably next section or next class. But ribosomal RNA makes up ribosomes, and that's a really important molecule within the cell. Sorry, a structure within the cell. That's where proteins get synthesized. So if something goes wrong in making new ribosomal RNA, probably the cell's not going to survive. So basically, the mutation rate is very, very slow for ribosomal RNA. It's highly conserved, is another way that we say that. So they reasoned that that would be a good molecule to look at to understand evolutionary relationships. And sequencing ribosomal RNA at that time was a really laborious process. They had to run gels. It took forever to do that. But they came up with some pretty interesting data. They recognized that no matter, when they did lots of different organisms, all of life actually fell into 3 different groups. Okay, so they had ribosomal RNA that was very 3 distinct sequences of ribosomal RNA. And that was surprising. They thought they were going to find 2 groups, prokaryotes and eukaryotes. And actually they found 3. So that led to the formation of what they argued should be a whole nother layer of taxa. So we talked about that kingdom, phylum, order, etc. So today we actually have a taxa above that that we call domains. So there's 3 domains: the bacteria, the archaea, and the eukaryota. Whoops. Sorry, let's call this eukarya. I knew that didn't sound right. This is a Y. Eukaryotes. Okay, so the bacteria and the archaea are what we we still call them prokaryotes, but they're prokaryotes, and of course all the eukaryotes are in the eukarya domain. Okay, so the work that they did led them to proposing this phylogenetic tree of life. And unfortunately, your textbook, it doesn't have this in your textbook, so I just grabbed this off the internet. But this is our accepted kind of understanding of sort of the correct way or the correct phylogenetic relationships. So if you look at this, here we are, over here, we're the animals. We actually share a closest common ancestor with the fungi. Okay, kind of surprising, pretty closely related to plants there as well. But all of these other groups in the eukarya are probably not very recognizable. Most of them are what we would call protists, or what we used to call the protists. So we'll talk more about those a little bit later. And then there's the bacteria and the archaea. So we're not going to talk a lot about archaea in this class. We've traditionally thought of them as being prokaryotes that live in more extreme environments. So for instance, the halophiles, they live in places where there's a really high salt concentration, like the Great Salt Lake or the Dead Sea, really hot places. So Thermoproteus, for instance, lives in hot springs. For some reason, I don't know the story here. This is an actual organism, Thermococcus cellar. I'm not sure why they have a particular organism there rather than a group of organisms, but all of these taxa are correctly actually called kingdoms now. Okay, so that's very different from 50 years ago, what we used to call that. Within the archaea, there are organisms that produce methane. So a lot of kind of bizarre things we now know that there are archaea as part of our normal microbiota. So you probably have some archaea in your body. It's not just extreme environments. You might have some in your water heater at home, or your hot water heater. But we don't know of any that cause disease. So we don't pay a lot of attention to them in this class. I'll mention them every now and then. Of course, we're going to put most of our focus on the bacteria. Again, a lot of these taxa are not necessarily even familiar to me. Most of the organisms that we use on a regular basis are either in the gram-positive group or this proteobacteria group. But there's photosynthetic bacteria. Of course, there's ones that live everywhere. Interesting thing here, look at this closest common ancestor. You would think that the archaea would be on the same size as the bacteria, but that's not what we see. And that was another really surprising thing that Wose and Fox found. So as we think, again, all these organisms in each domain, they share characteristics as well as common ancestors. If you kind of look at that group of archaea, now I'm going to kind of flip to chapter 11, where we talk a little bit more detail about things that we use for classification. If you think about comparing the bacteria and the archaea, and really, I took this information right from the book, but ideally we would have the eukarya in their own little box there as well. But when we look at that group, the archaea, they really have some things that are really you only find in that domain. And then a lot of things they either kind of borrow from the bacteria, or maybe they're a little bit more like the eukarya. One really distinct thing that we always use, and we're going to look at the structure of this, hopefully we'll get to it today, this structure of peptidoglycan. We only find that in the bacteria domain, right? So archaea, they have cell walls as well, but their cell walls do not ever have peptidoglycan in it. The eukaryotes that have cell walls, like the fungi and the plants, they do not ever have this structure peptidoglycan in it. So this is unique to the bacteria. If you look at some other structures like the cell membrane, for instance, we don't go into this in too much depth in this class, but the structures of the membrane lipids for bacteria are very different than the membrane lipids in the archaea, which are even different than the ones that we have in our cells. A lot of times the archaea will kind of borrow their metabolic processes from bacteria, and then some more of their genetics, they borrow a little bit more from the eukaryotes. So for example, in protein synthesis, the archaea use that AUG start codon, like the bacteria and eukaryotes, but it codes for methionine like the eukaryotes, where the bacteria, that start codon when they're putting their proteins together encodes for formylmethionine. So just kind of little details here and there, but really the peptidoglycan is kind of that big thing. And I know we haven't talked about that yet, but we're going to look at that structure a little bit more closely. And then thinking about the things that we have always used to put bacteria into groups, some of the things that we can easily see, maybe under a microscope or not even needing a microscope in some cases. Just some general terminology to do with the morphology of bacterial and archaea cells. The shape. So again, these are just kind of terms that you got to know. If they're round cells, we refer to them as coccus, plural cocci. And if they're rod-shaped, then we refer to them as a bacillus or plural bacilli. Okay, so if you're in lab, you're going to see a lot of cocci and a lot of bacilli. Pretty much not much of any other morphology. If we had a cell that was kind of part way, not perfectly round, not really rod-shaped either, we would call that coccobacillus. Okay, and each species is going to have, you know, a certain morphology that it almost always is. Comma-shaped organisms, we call those vibrios. And sometimes the morphology of an organism that will be incorporated into its genus name. So Vibrio cholerae, the organism that causes cholera, okay, it has a Vibrio morphology. This pleomorphic, that just means that the shape of the cell is not set. For instance, organisms with no cell wall, they're considered pleomorphic because it could be lots of different shapes. Occasionally the shape can change depending on the environment, but that's not terribly common. And then there's just lots of weird stuff out there. There's square-shaped bacteria, star-shaped. There's kind of some bizarre things. The helical bacteria, there's two kinds of helical, the spirillum and the spirochetes. They move in a corkscrew-like fashion. Okay, so they're really kind of cool. I'm going to show you some animations of each. Those are easy to kind of mix those two up. The spirillum, these are a little bit more, the morphology is a little bit more rigid in terms of the body, and it has flagella at the ends. Okay, so it will usually have like a tuft of flagella at one end. You can't see that in this picture here. And the spirochetes are a little bit more flexible, and they have their flagella are internal. Oops, an A at the end there. So we're going to look at the different kinds of flagella hopefully today, but it has a flagella that runs all the way down its body, and it's actually enclosed in a membrane. Okay, so again, telling those two different morphologies apart, spirilla and spirochetes, just little animations here. It's always fun to see this. This one is of spirilla. Let's see. Come back. There it is. Okay. So let's see. You can get lost for a long time on YouTube looking for stuff like this. Let's see. Go. Okay. So again, kind of the body shape is, it doesn't really, oh, where are you going? Okay. Let's see. Oh, I didn't want to go that far. I guess that's mostly what I wanted to show you there. But again, it's fairly rigid, but you could before you could kind of, you can see it moving in a corkscrew. Let's look at the other one here. This is, so think about reviewing our kinds of microscopy. Any guesses what kind of microscopy that spirilla one is? That picture there. 3D image. This is actually a scanning electron micrograph here. That might be hard to tell. It's helpful if they tell you the units. You know, so if it's really small units, you know that it's going to be scanning or transmission electron microscopy. This one over here is dark field. And so I wouldn't maybe necessarily know that from looking at it to tell the difference between fluorescence and dark field, because both of those are going to have a dark background. But based on, well, we'll take a look at this. If you're going to, remember, see living organisms moving around, remember you can't stain them, because the staining always kills them. So we know that this video for sure is going to be dark field microscopy. And again, black background, Phase contrast doesn't kill. You don't need to stain using phase contrast as well, but that's going to be more of a grayish background. So here, longer spirochetes, they've got the internal flagella and kind of longer bodies that are a little bit more flexible. So, all right. Okay. All right, so kind of a first hint that kind of helps us maybe put organisms in group as their body shape. Also, we look at very carefully how those cells tend to arrange themselves. So how they arrange themselves is based on two things. Whether when they divide, they stay attached. So they stay attached when they divide. Okay, sometimes they do a little bit, maybe for a short period. Sometimes they stay attached for long periods of time. And the plane or planes of division. Okay, so when bacteria cells divide, one cell becomes two. Does that always happen along the same plane, or can that happen, you know, in different planes, and it can kind of stay stuck in kind of different directions? So again, there's just kind of some terminology that I want you to be familiar with. Here's a cell that divides only in one plane, okay, and this is all cocci in this graphic. Okay, so here we've got diplococci, usually pairs of cocci, because the cell, again, divides in one plane and then probably only stays stuck together, not for very long. So when each of those daughter cells divide again, they come apart. Sometimes the cell will divide in one plane, but then they stayed stuck together for a long time and form chains. Okay, so again, sometimes the name of an organism will give us a hint as to either the morphology or the arranged cell arrangement. Streptococcus, those are round cells that tend to form chains. Okay, that prefix strepto means chains. How about a cell that divides in two planes and then stays stuck together? That's gonna form packets of 4 that we refer to as tetrads. 4 cells, that makes sense. And then if we add a third plane, it's going to be more 3D, more of a cube arrangement. And we have a special term for that called sarcina. So when you look through a microscope at cells that are either making tetrads or sarcina, in practice, it's kind of hard to tell them apart, because it's kind of hard to tell if they're, you know, when you're looking at a 2D image, whether you've got a cube or just a group of 4. Sometimes you can. And then some cells divide in many planes and they stay stuck together. So they form clusters. That Greek staphy, that's Greek for clusters. So staphylococcus, round cells that tend to form grape-like clusters. All right, so again, we see those arrangements under the microscope, and then that gives us a little bit of a hint, maybe, as to what they might be. Less terminology to do with arrangement of bacilli, because our bacilli don't divide in many different planes. Our rod-shaped cells, they will only divide right there in the midpoint, okay? They would never divide long ways like this, okay? We don't see that. Okay, so those cells, they get longer and longer, and then they pinch off the septum in the middle and divide. So we could get diplobacilli, pairs, streptobacilli, chains. Anthrax makes streptobacilli like that. And then really the only other thing to mention here is that sometimes when bacilli divide, they kind of hinge a little bit. They stay attached kind of just at one spot, and then they hinge open and around and can form this arrangement that we call palisades. Okay, so where that term comes from, if you've ever been to like Fort Mackinac up on Mackinac Island, the fence that goes around the fort, right? Lots of logs lined up alongside to alongside. We call those palisades. Okay, so that's when we get rod-shaped bacteria kind of lining up like that. We call that a palisade arrangement. And again, it's not because they split long ways, it's because they kind of hinged like this around on each other. Okay, so that's a little bit harder to pick out when you're looking at cells, but sometimes you see that. Okay, what are the things are we looking for to classify organisms? A very common thing to look for, it's not common among bacteria though, is the formation of endospores. And we're going to talk about this a little bit more later, but there are only a few genera that can make endospores. So it's not common, but it is very kind of distinctive because it is a kind of a survival mechanism. Okay, so basically only a few genera have the ability to form endospores, and that just means they can kind of hunker down into almost, you can think of it as kind of like a seed, like a dormant structure where they're not metabolically active anymore, and they can persist that way for years and years and years. They usually will do this when conditions are poor. Okay, so they encounter some environment that's not conducive to them growing. So there's not very many nutrients, or the temperature is too high, or the pH is off, or it's dry. Okay, you can think of lots of things. And then when conditions improve, they can germinate and become metabolically active again. So if you're in lab, you're going to get a chance to do a spore stain in a couple weeks where the spores take up one dye. That's the small greenish colored. They're not round. They're really kind of little ovals. And then metabolically active cells, the vegetative cells, will be red. I won't ask you about the staining, but just again, if an organism is a spore former, then that's a very distinctive thing about it. We can use that for classification. So what things have we talked about? We've talked about the shape of the cell and the cell arrangement, whether the organism is able to form spores or not. And we're going to talk more about this soon, the Graham reaction. Okay, we mentioned that Hans Graham made up this Graham stain to help us be able to visualize cells better. And that's what we call a differential stain. So it stains some cells one color and some cells another color. So it's usually the first question we ask when we're trying to identify an organism. Is it positive for that Graham stain, or is it negative for that Graham stain? And that makes a difference as to, what kind of antibiotics we're using if we're trying to kill it, for example. So Graham reaction, we'll talk more about what determines whether it's Graham positive or Graham negative or not soon. Also, the colony morphology. So remember, colonies are those discrete packets that grow up on media that we can see with our eyes, right? Millions of cells within one colony. So there's, a lot of colonies, they're just like little white circles, right? But the closer that you look at them, and I can't remember, I might not have posted this because I don't want you to memorize all this terminology. I don't have it memorized. But actually, when we look at colonies closely, we start to notice things like they might have a different shape to them, or a different margin, a different edge. The center might be kind of cropped up, or it might be very flat, different sizes. It might seem dry or shiny, different colors. So there's kind of all different kinds of colonies. And when you work with a certain organism for a while, you get really good at, oh yeah, I can tell that it's that organism because just by looking at it. All right, so that can help us identify organisms as well. And now we're kind of bouncing back to chapter 4 here to finish up thinking about classification. But other than those things that you can just tell by looking at about bacteria, we use a lot of other things like their biochemical characteristics. So if we were looking at a larger organism to try to classify it, we would look at its structures. Oh, does it have gills or lungs, for instance? All right, we can't do that very easily. But so for bacteria, we use things like, well, what nutrients can it metabolize? Lactose is a big one. There's a lot of variation there. Some bacteria can metabolize it, have the enzymes to do that. Some of them don't. So that's a thing that we test for like 3 different ways if you're in 213 lab. So we give the cell some lactose, we put a pH indicator in there, and if the organism can metabolize the lactose, we look for acidic end product. So we look for a color change. Does the organism make a gas if it utilizes that nutrient? So there's different ways we can collect that. We might have a little tube inside there that looks for a gas bubble. We might have some other indicator. In this case, we're looking for a gas, hydrogen sulfide, that turns a black color. Just an example there. Maybe we're going to test for a certain enzyme to help us, understand what groups an organism should be in, or maybe even antibiotic sensitivity. I don't think that's as commonly used for classification, but we got little antibiotics in these discs here, and then we're looking to see if the organism is inhibited by that antibiotic. That's something that we'll talk about in here later in the semester, and you'll do it in lab. So lots of things we use. We're moving more and more now into using even more advanced criteria like sequencing. Like I talked about the ribosomal RNA or maybe DNA, or using antibodies to be very specific and understand structures. So where can we find out all the information that we have about any one bacterial species? There is a go-to source for that. It's called Burgess Manual. It comes in about 5 different volumes. You can access it online too, but if you want to know everything there is to know about a certain organism, then there is a way to look that up. All right, so questions. So I have finished up what I'm going to cover about chapter 1, 6, 4, and 11. All right, so that chapter 11 is kind of a survey of prokaryotes, and I just kind of dipped a few pages into that. So ready to move on and think about structures of prokaryote and eukaryotic cells. All right, questions? Don't be afraid to stop me if you have questions. All right, so this is really, again, important material because we're going to be kind of referring to a lot of structures that we talk about here for the rest of the semester. So good basic information. I'm mostly going to talk about prokaryotic structures first, and then a little bit more briefly talk about eukaryotic ones. But at times it's helpful to just kind of compare them both at the same time. So we'll first talk about, this should be a review, what's the difference between prokaryotes and eukaryotes. We're going to spend a lot of time on prokaryotic cell wall structures, not so much on eukaryotes, not a lot of time on the membranes, a little bit of transport stuff, things like that. Another structure that both prokaryotes and eukaryotes can have are flagella, and there's quite a bit of differences. Other than flagella is useful to move the cell, everything else about prokaryotic and eukaryotic flagella is different. And then we're just going to really touch on this briefly, some of the organelles in eukaryotic cells. I kind of assume that you've had that at some point before, and we're only going to kind of focus on the ones that we're going to talk about later on when we get to our immunology portion. So, all right, so a little overview between prokaryotic and eukaryotic cells. Of course, the prokaryotes, like we just talked about, in two different domains, the bacteria and archaea domain. I remember what I was going to talk about. When we were talking about the domains, I had that whole tree of life. Where do you think viruses would fit into that? We haven't mentioned viruses yet. Would we have viruses on that tree of life? What do you think? So aren't they considered not alive because they're acellular? Yes. And remember, the basis of that tree of life was ribosomal RNA. Viruses do not have ribosomal RNA, so they wouldn't be on there at all. And we'll talk more about whether we consider viruses alive or not alive a little bit later, but most scientists would say not, but there's still room for argument about that. Okay, so sorry to bop back there. Sometimes I mean to mention something and then I forget. So prokaryotes, defining thing here is that they lack a membrane-bound nucleus. Now they have DNA, and that DNA is all usually in one part of the cell, but there's not a membrane that goes around that. Okay, and in fact, they don't. Prokaryotic cells don't have any structures that are bound by membranes. Okay, inside they have a cell membrane. They just don't have organelles like eukaryotes do. Size, how big are they? Usually on the order of about 1 micrometer, 1 to 2 micrometer, or maybe even a little bit less. Okay, there's always variation. So for example, here's a scanning electron micrograph. Look at your units here, 1 micrometer. Okay, so diameter here, on average about 1 micrometer. So if you compare that, here's the protozoa here. Look at the units here, 40 micrometers. Okay, so that eukaryotic cell is 10, 100 times bigger than a sample prokaryotic cell. All right, so again, the opposite there. If we look at eukaryotic cells, they do have a membrane-bound nucleus, and they do have lots of organelles that are bound by a membrane. So what kind of cells are eukaryotic? We're going to talk about these next week, hopefully. Algae, fungi, plants, animals, protozoa. Okay, every once in a while somebody will get confused when I say, is it difference between prokaryotes and eukaryotes? Don't confuse that with what's the difference between plant cells and animal cells. Okay, animal cells don't have a cell wall. Plant cells do. Okay, some of these eukaryotes do have cell walls, some of them don't. Okay, so size somewhere between 10 micrometers to 100 micrometers if you look at the diameter. Okay, so one side of the cell to the other, and of course they're not all perfectly round. It would be different dimensions, but okay. All right, so starting with the outside of the bacterial cell and working in. And again, I'm talking specifically about bacteria. There are some specific structures for the archaea, but we're not going to worry about those. First thing you come to that most bacteria have on the outside is some kind of glycocalyx. Okay, and that's basically a general term for a slimy substance secreted outside of the cell. If it's really rigid and tightly bound to the cell, we would consider it a true capsule. Okay, so we can say tightly bound. If it's a little bit loosely organized, we would consider it a slime layer. Okay, so a glycocalyx can be considered a true capsule, or if it's a little bit thinner, loosely organized, we might just call it a slime layer. It's really a continuum. Like sometimes it might be hard for us to say, well, would we consider it a capsule or a slime layer? Typically going to have a polysaccharide composition. So sugars. So lots of repeating subunits. There are exceptions to that. There are a few organisms that have a little bit more protein in their glycocalyx, but more often carbohydrates. And that's important when we think about trying to visualize it under the microscope, because a lot of our dyes, they stick to structures based on unlike charges. And these polysaccharides that make up capsules typically don't have any charge. So it's difficult to get a dye to actually stick to the capsule. More about that in a minute. I'll show you an example of another, the way that we stain it in class here. It's important also to understand that most cells produce some kind of a capsule or a glycocalyx, whether you consider it a capsule or whether you consider it a slime layer. Most cells produce it because the advantages are so great to doing so. So what is its function? One, attachment. Could be attachment to other cells or some kind of structure. Okay, so that's capsule is kind of sticky, helps it adhere to other cells, makes biofilms, large masses of cells. Also enables some cells to stick to things like our teeth. Okay, bacteria in our mouth need to be able to make that slimy capsule so they stick to our teeth instead of being washed down into our digestive tract. Okay, so bacteria that can do that, they contribute to our tooth decay. Another function helps the cells resist drying out, desiccation. Okay, so a bacteria out maybe on a surface of a desk, for example, isn't going to dry out as quickly as it has this moist capsule around it, stay viable for longer. And another important function is that it helps cells resist phagocytosis by our white blood cells. All right, so if you have an organism that's trying to establish an infection in our bodies, it's going to likely be a lot more successful because our kind of first line of defense, our white blood cells, are going to have a harder time phagocytizing it and getting rid of it. Okay, now eventually we have kind of smart weapons called antibodies that would be able to help white blood cells phagocytize that, but at least in the initial stages of infection, helps an organism become established. Okay, so most organisms do make some kind of a capsule. This graphic from your book is kind of similar. It's not exactly what it looks like when we try to do this in lab. If you're in lab, you'll get to do a capsule stain next week. But the capsule here is this unstained part around the cells. All right, because it's hard to get dyes to stick to the capsule itself, we stain the background instead, as well as the cell wall. And then that unstained part around the cells is actually the capsule. Okay, so we need a special stain to see that, otherwise we kind of forget that it's there at all. So, okay, so working our way into the cell, next we come to the flagella. All right, so we haven't come to the cell wall or cell membrane yet. The flagella is outside of that. And of course, the main function of flagella is to help move the cell in a purposeful way. All right, so help it to move towards some attractant, whether it's a nutrient, whether it's sunlight. Okay, and not all cells have flagella. For instance, I don't think there's any cockeye-shaped bacteria cells that have a flagella. There may be some exceptions somewhere, but I haven't run across any. So not all bacteria are what we would say motile. Typically, whether they're motile or not depends on whether they have flagella. All right, so if we look at the structure of flagella, we're gonna show you a graphic in a minute that's in detail. But the flagella is composed of a filament, a hook, and a basal body. Okay, and that filament or filaments, a cell can have lots of flagella, is composed of a single protein flagellin. Let's see if I can spell this right. Okay, so that's pretty intuitive. Flagellin makes up flagella, and we could look in detail at the structure of that, but we're not going to take the time to do that. And then the basal body is basically what anchors the flagella into the cell wall. And the hook is just kind of an attachment there. All right, so if we look at the graphic that the book shows you, again, a little ahead of ourselves because it shows two different ways that it's anchored into the cell wall. We haven't talked about the cell wall structure yet, but the top one is here is how the flagella would be anchored into a gram-positive cell wall. Again, differences here with the basal body, but you see the filament, this hook region, and then again, this is the basal body. You can see it talks about rotating. Okay, so movement is due to rotation. Okay, if the organism only has one flagella that moves one way, it will go in one direction. If it moves the other way, it will go in the other direction. Pretty simple. The bottom picture here is just differences in how the flagella is anchored into a gram-negative cell wall. And there's just a lot more layers in a gram-negative cell wall. And again, we're going to pick that apart in a little bit. Okay. So can different organisms have different arrangements or kinds or numbers of flagella? Yes, they can. All right, so, and of course we have some terminology to describe that. If organisms have one flagella, we call them monotrichous. Okay, mono meaning one. Okay, and if an organism only has one, it's always going to be at one end or the other. So here they say single polar flagellum. Polar means at one end or the other. You're not going to have a flagella sticking out the side of a rod-shaped organism like that. We don't ever see that. The other very common cell arrangement we call paratrichous. Sorry, flagella arrangement. Paratrichous, okay. And that means we have flagella all over the cell. Okay, many of them all over the cell. All right, so here the top picture, it's kind of hard to see why in the room here, that picture, but this bacterium has lots and lots of flagella all around. And that's actually a very common flagella arrangement. E. coli has that. We kind of forget when we get used to seeing cells in the lab that most of them do have flagella because we can't see the flagella unless you do a special stain for it. So with light microscopy, it's very difficult to actually see the flagella. Peri here, peri means around. So if you were measuring the perimeter of a room, you would measure the distance around. Okay, so that knowing your prefixes there can kind of help. We also see sometimes organisms that have a tuft of flagella at one end or the other. And I won't burden you with any additional terminology there, but okay. So we saw that in the sporillum of a tuft at one end or the other. The other thing that we see is an axial filament, and I mentioned this before. This is our spirochete morphology. This is a flagella that is enclosed in a membrane. Okay, so the other flagella do not have a membrane around them. They're considered outside of the cell. But this is a specialized flagella, the axial filament, that's actually composed of many endoflagella. Okay, so you can see that here, the cross section, this structure on this top there, that's the flagella that's right up against the spirochete cell or body. And within there, all these little dots, those are endoflagella. Those endoflagella are all spinning, and that's what makes the cell move in this corkscrew-like motion. So, you know, give you an examples of organisms that have this morphology. This is Treponema pallidum. That's the organism that causes syphilis. So organisms that can move in this corkscrew-like manner helps them kind of penetrate through maybe thicker, more viscous materials here. The organism that causes Lyme disease is also a spirochete, in case you're interested. The motion of the flagella, again, it depends if you've got an axial filament, of course, but if you've got one flagella or many flagella, the motion can be a little bit different. But if an organism has many, if it's a paratrichous arrangement, then if those flagella are moving counterclockwise, spinning counterclockwise, then they are going to move in a coordinated way. So we call that a run. Okay, otherwise they're going to move in a straight line. Okay, so we see that when the flagella move in one direction for the paratrichous, you can see that right here. They're moving counterclockwise and they're coordinated. They're moving all together. But in this example here, the attractant is up here. So this cell can sense that it's not getting closer to the attractant. So it needs to stop and kind of head off in a different direction. So it does that by moving the flagella into clockwise direction, and that will cause it to have a tumble, meaning that it will kind of spin around in every direction, and then it will run again and kind of go off in another direction. So you get these series of runs and tumbles till it eventually will make its way towards some attractant. So I think there might be an animation as part of your homework. I found a few here that I thought were good ones. So let's take a second and look at these. Let's see. This one is just a drawing. So if you hold up the shot, you hear the nice background music. So it's kind of spinning around, opening and tumbled right there. Okay. I think these are are better ones here. Okay, so one thing to understand is, like I said, not all bacteria have flagella. They don't all move purposely. If you're looking at cells like here under a light microscope, when they're just kind of flowing with medium, that's not motility. That's what we call Brownian motion. They often will be kind of shaking a little bit, but just kind of going with the flow. That's not really true motility. What we would be looking at here, and again, these are kind of small to look at, but if you kind of focus on one, you can see how some of them are moving purposefully. They'll dart off in one direction. Sometimes you'll see them kind of spin around and then dart off in another direction, if you kind of watch it for long enough. We can do this in lab if we do a wet mount. So we can use our bright field microscopes and just kind of put a cover slip on there. And if you get the light just right, you can see it. These show up a little bit better. There's a number of cells that are just kind of dead or stuck to maybe the cover slip or the slide. And then you see some other ones buzzing around there. So that's kind of fun to watch. Again, most of the time they're paratrichous flagella going in runs and tumbles. Okay, and I think that third one is just really more the same of what that other one was. So if you want to look at that sometime, if you're interested. Okay, so questions about flagella, kind of fun, I think, to think about those a little bit. Other structures that are kind of similar to flagella, but they are not purpose, their purpose is not for the movement of the cell, are fimbriae and pili. And we think primarily these are found in gram-negative organisms. I know I haven't talked about what that means. We'll get to that in a minute. But both of these structures have the same composition. They're composed of a protein called pilin. Okay, so the difficulty is, well, if it's made of the same thing, why do sometimes we call them fimbriae and sometimes we call them pili? There's not hard and fast rules about that, and sometimes we argue about what to call them. In general, if these, I don't want to call them appendages, if these structures, hair-like structures, if there are many of them and their function is mostly for attachment, then we tend to call them fimbriae. All right, so these long hairs here, those are flagella. So what we're looking at here are these short kind of hair-like structures around the cell, primarily so that the cell can attach to some other structure. Okay, so for example, E. coli has fimbriae. It's very important that they have fimbriae because we all have E. coli in our large intestine, and the fimbriae help the E. coli adhere to the epithelial cells of our large intestine. If they couldn't attach themselves to that, then they would wash out with our wastes. So a very important structure for a lot of organisms to stay where they want to be, to adhere to certain cells. Sometimes we call these structures pili. And normally we would do that if the cell only has one or two of them, and they're a little longer, and their function is to facilitate conjugation. So I know we haven't talked about conjugation, but that's a kind of horizontal gene transfer. So it's when one cell actually shares some of its DNA with another cell. Okay, and they can only do that if they can extend a pilus to another cell, and that pilus is kind of drawn in, so the cells get drawn closer to each other. A pore opens up, and then they share DNA. So they don't share DNA through the pilus, but it's just a way to draw the cells close to each other. And this, I think they fixed this in a newer edition of the book, but this is not, the one on the left here, this is a scanning electron micrograph. This is also a scanning electron micrograph. Remember, a transmission electron micrograph, how would that look different? It would be a cross section. Okay, so that's where we slice the cells really thin, and then what we view as a cross section. So that definitely doesn't look like a cross section to me. All right. So again, these are structures some cells have, mostly ones with a certain kind of cell wall, and it can just aid their survival. All right, so let's get to the cell wall. An enormously important structure that we're going to spend a lot of time on. Don't worry, there's not a lot of things to talk about inside of the cell wall, so we're going to spend a little bit more time with that. Purpose imparts the shape of the cell, okay, and strength and protection especially helps the cell withstand changes in solute concentration. So you may have done this. I can remember years ago when I used to teach Bio 120, we would do this. We would put some red blood cells on a slide, and then you would add a solution that was hypotonic, had low solutes, and try to wick it across under those red blood cells. And what would happen? Bam, the cells would burst. Okay, because bacteria cells have a cell wall, if you put them in a concentration where there's very low solutes, water is going to rush in, but because of that cell wall, it's not going to burst. Okay, I think we did the same thing with a plant cell. Okay, they're going to crenate, when they in a hypertonic solution, but they're not likely to burst. It would take a really huge difference in solute concentration. So really, really important for bacteria cells to have that protection. Composition. Several different kinds of cell walls, but what they all have in common, all bacteria cells, like I alluded to, they have the structure of peptidoglycan. Okay, again, that is in all bacterial cells with a cell wall. Now, there are a few bacteria that don't have a cell wall, but most of them do. And if they do, they all have peptidoglycan in it. Okay, so what is this? If we pick this word apart, glycan, okay, that root should make you think sugars. Okay, so it's actually composed of two alternating sugars. Okay, so this is the sugar portion. N-acetyl glucosamine. Yeah, sure I spelled that right. Oh, I knew that didn't look right. I gotta spell that right. There we go. Had the Y in the wrong place. N-acetylglucosamine. Okay, we abbreviate that oftentimes and call it NAG. It has alternating sugars, N-acetylglucosamine and N-acetylmuramic acid. Okay, and that we abbreviate that NAM. Okay, so alternating sugars. And if we look at, I'm going to come back to this in a second. If we look at the structure of those sugars, I don't think I posted this, or if I did, certainly don't memorize this. The structure is pretty similar to glucose. Okay, so this just highlights, the only difference that N-acetylglucosamine, only place it's different from glucose is in this gray portion. And then N-acetylmuramic acid is very similar to N-acetylglucosamine. Only difference here is this portion in orange. So again, they're sugars similar to glucose. And then this peptido root of the word makes you think peptide. So yes, there are also amino acid cross bridges. Okay, so if we look at a schematic of the structure of peptidoglycan, this is what your book comes up with. Okay, so we have these alternating sugars, NAM, NAG, and then layers of those, and then we've got these little circles. Those are the amino acid cross bridges. Okay, so it's a pretty rigid structure. Kind of think chain link fence would be a good analogy. Okay, so remember the cell wall is not, it's not the gatekeeper. That's the job of the cell membrane. It decides what enters and leaves. If you think about a chain link fence, anything that's small enough to fit through there is going to flow right through. All right, so it's not selective. That's the word that I'm looking for. Anything can fit through there, but it does provide a good, again, good protection. So before we get to the difference between a gram positive and gram negative, I want to talk for a minute about the growth of a cell. And your book doesn't talk about this, but I think it's important to mention it because later on, when we talk about antibiotics, this becomes really important. So when the cell grows, right, it gets bigger and bigger until it splits. So the cell wall has to be able to grow. So it has to be able to add in new NAG and NAM sugar units. Okay, so the way it does that, it's going to cut these bonds between them and then add new ones. So it has to cut those bonds between the sugars. It has to cut these bonds, these amino acid cross bridges, before it adds new sugar units. Okay, so that all has to be very carefully orchestrated. Okay, the autolysis, these are enzymes that cut the bonds between the sugars. Okay, so those sugars are made inside of the cell, transported through the cell membrane and put into the cell wall. And then it's the job of other enzymes to reseal those bonds, between the sugars and amino acids. And transpeptidases reseal the bonds between the amino acids. And it's the job of other enzymes to reseal the bonds between the sugars. I just didn't want to burden you with too much. I wanted to keep it as simple as possible. Okay, so this is the best graphic that I could come up with. Again, they don't talk about this in the book, but those sugars are made inside of the cell, in the cytoplasm. They are shuttled through certain channels. We don't worry about what those are called. And then they are, again, the autolysis have to cut these bonds and the new sugars are put in there. This TP, that stands for the transpeptidases that reseals those amino acid bridges. All right, so what happens if something inhibits the transpeptidases? Then you've got a cell wall that's not going to be quite as intact. It's going to be a little bit fragile, and it's going to be susceptible to bursting due to osmotic pressures. Okay, so that's actually what happens when a bacterial cell is in the presence of a certain kind of antibiotic. All beta-lactam antibiotics like penicillin, they actually bind to the active site of the transpeptidases, and then those amino acid cross bridges don't form again. So you've got this cell wall that, again, is not in good shape. And this is a time lapse, movie of what happens when you add some antibiotics like penicillin. Okay, they add some sound effects there of the cell popping like a balloon. It happens a little bit more slowly than that, but it's not the penicillin that actually kills the cell. It just weakens the cell, and then osmosis does its thing. So that's kind of a preview of what's to come when we get into talking about our antibiotics. Okay. All right, so all of our bacterial cells have peptidoglycan, but our gram-positive bacterial cells have a whole lot more. So what's unique about this special kind of gram-positive cell wall? They have, we'll say, lots of peptidoglycan. Somewhere between 40 to 80% of the cell wall by dry weight is peptidoglycan. Okay, so a mnemonic that may help you if you think about gram-positive cells, lots of peptidoglycan, gram-negative, very thin layer peptidoglycan. Okay, and that's really the main thing, that when we stain them, the gram-positive cells, because there's so much peptidoglycan, that holds on to the dye really well. Gram-negatives, it doesn't hold on as well, so it gets destained, decolorized really easily. And then the gram-negatives take on the color of the counter stain that we add. All right, so you are going to get to do that in lab if you're in lab next week. So I'll let you talk more about gram staining in lab. The other thing that's unique to gram-positive cell walls is teichoic and lipoteichoic acid. Okay, and I'll show you a graphic of those in a minute. We think that those molecules help determine the cell shape. And they may have something to do with regulating cell division. It's not totally clear, but again, this is unique to the gram-positive variety of cell walls. Okay, so if we look at the graphic here that's from your book, I think it's helpful sometimes, instead of just kind of looking at them, to go through and kind of label it yourself whenever you can. So here's the cell membrane here down at the bottom. We haven't talked about that yet. But this whole pinkish, Krabby Patty type looking thing, that's the peptidoglycan. Okay, there's a ton of it. And then these only other structures that we see in here, we call it teichoic acid if it doesn't anchor into the cell membrane. And the ones that do, we call lipoteichoic acid. Okay, so again, it anchors into the cell membrane. Okay, so it's really fairly straightforward, fairly simple. There's not a lot of other things that we're going to talk about in there. Okay, I think that's a good place to stop. We're going to discuss in more detail the gram-negative cell wall next time. It's a lot more complicated, but good. So we'll try to finish up chapter 3 on Monday. Try to catch up on your homework. And one last chance, this doesn't look familiar to anybody. Somebody left this in lab. That's all our classes now. Okay. All right. Have a good weekend, and we'll see you next week.